It is a great pleasure for me to introduce Professor José Obeso, not only because of his distinguished career as an expert in movement disorders, but he is especially a good friend of mine. We started together in the field of movement disorders many years ago, and he was a good friend then, and he's still a good friend now. Dr. Obeso, who is a graduate from the University of Navarra, had his main instance of approaching the field of movement disorders when he had the chance between 1980 and 1982 to work as a researcher under the mentorship of Professor David Marsden, one of the founders of our society, in his lab in London. Dr. Obeso is currently director of the CINAC at the Hospital Puerta del Sur in Móstoles, Madrid, Spain, and full professor of neurology in the CEU San Pablo University in Madrid. He has made significant contributions to the field in the areas of pathophysiology of the basal ganglia and surgical treatment of Parkinson's disease. He's also the chief editor of a signature journal, Movement Disorders, the leading publication in the field of Parkinson's disease and movement disorders. He's also an honorary member and invited professor of several organizations and institutions, such as the Colombian Neurological Association, the French Neurological Society, the Bolivian Neurological Society, and he's a professor of the University of La Habana in Cuba. Dr. Obeso, he's going to talk about basal ganglia and pathophysiology, puzzles, paradoxes, and clues.
Thank you very much, Oscar. Good morning. It's a big honor and pleasure to have been appointed to give this lecture today. Let me see how the lecture will be dealing with, as it is customary, with the name of the lecturer, Professor Marsden. In this case, I'm going to take the liberty of being a little bit in more detail, since Marsden was my mentor, my professor, my friend. Also because the tight work we did together on the pathophysiological models of the basal ganglia, I'm going to quickly revise some of these aspects, as well as the classic paradoxes derived from the model, to allow more time to my current field of interest, coming from how findings in relation with basal ganglia pathophysiology may give way to some clues to understand dopamine cell vulnerability in the nervous system. Now, Marsden started his academic career with Professor David in St. Thomas Medical School with his master's degree on the substantia nigra in the cat. And this is how he apparently became involved, passionate by the basal ganglia. He did a quick clinical training period in Queen Square, and then at the premature age of 34, was appointed professor and consultant of neurology in the Institute of Psychiatry. This was the first chair of neurology across the river, and ever since, south the river in London became a trench in competition with the mecca of neurology north in Queen Square. The evolution of Marsden academic life was really rocketing. He was elected the youngest member of the Royal Society. He was among the 10 top authors in the 80s, and even after his death many years ago, he has this impressive ACE index among the 10 tops for all neurology and neuroscientists nowadays. But his real interest was in understanding mechanism, and Professor Pat Merton from Cambridge, who was a very in-depth and proper physiology, was his mentor from this point of view. They embarked to demonstrate that this stress reflex, the so-called low latency stress reflex, was using a cortical mechanism that caused tremendous polemic in the 70s and 80s, and they published many papers famous by the Marsden, Merton, and Morton authorship, giving all sorts of indications to prove that indeed this is a cortical mechanism. In London, he worked in the Institute of Psychiatry. This was his office. He spent many hours. It was difficult to arrive before or leave after Marsden in those years. The real good thing, if you stay there for long enough, is that at the end of the day, we could come out here, turn the right, and we'd go to this pub or this pub, and here is where the real good ideas on the best papers would keep written down here, and then off you go for the weekend to come back on Monday with a paper. In the 90s, when Marsden was at the peak of his career, this classic, what is now called classic model of the basal ganglia, was developed. Essentially, it shows that the striatal cells project in the direct pathway to the GPI and indirectly through the GPE, subthalamic nucleus, to the GPI. This is inhibitory and this is excitatory because of a double inhibition, and therefore it is supposed that both pathways maintain an equilibrium of control, one of facilitation, one of inhibition. Alan Crossman in Manchester played a fundamental role in developing and studying the subthalamic nucleus, but of course it were the paper from Alvey, Penny and Young, Malon DeLong, that had such a massive, major impact in the community. Essentially, the model just state that pulses in the output facilitate movement by disinhibition, discharges will reduce movement by increasing excitability on the pallidothalamic projection, and therefore from here, Anne Young and Penny and DeLong easily deduced that excessive activity could lead to Parkinsonism and movement inhibition, and reduced activity could lead to involuntary movement of chorea. And this is the essence of the model. The model was supported by enormous amount of data in the 90s. The MPTP monkey allowed to show that indeed there is hyperactivity in the subthalamic nucleus and GPI, recording neuronal activity. The GAD, the GABA enzyme, is increased in this nucleus and is reduced in monkeys after a subthalamic nucleus lesion. In patients, there is hypoactivity of the cortical premotor area and MSA. Before lesion, after pallidotomy, these areas became more active, indicating that their their excitability has been restored by a local action down in the basal ganglia to stop this excessive inhibition. And of course, we all know the tremendous impact of motor improvement with a surgery. More modernly and more recently, by optogenetic, it's been shown that indeed by selective activation of neuronal groups like these D2 neurons, medium spiny neurons in the indirect circuit in the striatum, can actually, as predicted, if this is a stopping inhibitory system, you will see that when these neurons are activated by the laser coming here, the mice stop, the laser goes, the mice move again, the laser comes, and the mice will stop again, showing that indeed the indirect circuit is very capable of stopping movement by activating that pathway. So with this in mind, one could somehow concede and conclude that the model fit with the notion that the motor circuit is normally involved in selection, facilitation of movement, and as a result, when this is faulting, bradykinesia is secondary to abnormal energization of neuronal activity in the motor circuit, a notion that Mark Hallett put forward in the early 80s. Remarkably enough, in my mind, and for the now almost 20 years the model has been around, there has been little attention to the fact that the two other major clinical features of Parkinson's disease, rigidity and tremor, have not really been understood and scrutinized from the same point of view. And if one thinks about rigidity, which is obviously a positive, a release sign in Jacksonian terms, you see all these changes in the Parkinsonian brain. There is increased motor cortical excitability, there is increased glucose metabolism by FDG in the motor cortex. The long latency stroke reflex, as shown before by Marsden himself, is increased, and in the MPTP monkey, there is increased neuronal cortical response. So this suggests actually that the Parkinsonian brain, and even for too tight motor features like bradykinesia and rigidity, they don't actually go together. This could suggest that there are two different mechanisms, and at one hand, we could have this premotor circuit going through, which is the one to the premotor areas and presupplementary area, which is hypoactive, while the primary motor cortex, basal ganglia, back to the primary motor cortex, also with a cerebellar influence, is actually hyperactive. And that may be, just may be, part of what is going on to explain rigidity. Similarly, tremor poses a tremendous puzzle here, because tremoric activity is present in many regions of the brain, in the basal ganglia, out of the basal ganglia, in VIM, which is a cerebellar receiving area projecting to the motor cortex, etc. So how these two circuits, the one where the dopamine deficit originating the tremor is somehow driving and recruiting this other abnormal circuit, which should be normal from the cerebello-thalamocortical pathway. This is really not understood, and is really a sort of puzzle that requires further understanding. One possibility is that there has been a recent description in my previous university, in a study that we did, showing that the subthalamic nucleus actually projects directly to the thalamus. So it could be that because the subthalamic nucleus is a very tremor related nuclei in the Parkinsonian brain, that could be a way whereby the basal ganglia interact with the thalamus and the cerebellar loop to integrate tremor activity. Of course, many of these puzzles and questions that one can pose from the point of view of the classic model are also part of our ignorance and the reduced information that we have. And in the 20-25 years since the model was first put about, there have been many changes. I'm going to summarize few for you. First is this hyperdirect pathway that from the cortex to the subthalamic nucleus that is being histologically and physiologically proven, and in a recent paper published in Movement Disorders Journal by Professor Quartone, you can see here this beautiful tractography showing the fibers arriving into the subthalamic nucleus. Also, the same group have shown projection from the cortex to the GPE, to the GPI, and these beautiful pictures also. I think this is in the March issue. We made it a cover because the figures are really very impressive. This requires, though, a careful histological confirmation. Histologically, it's being confirmed in the mice that the GPE projects back to the cortex, and further work could eventually be proven that there is this loop, cortico-GPE cortex, which could be a new loop and a new way whereby cortex and basal ganglia would be interacting and modulating movement, behavior, and emotions. Interestingly also, the GPE is no longer considered a go-through structure. The GPE projects to the thalamus, as André Perrin showed many years ago, also project to the GPI directly, and back through these archipallial neurons to interneurons in the putamen, and is modulated by these dopamine, extraatrial dopamine axons to the GPE, GPI, and STN. Therefore, the basal ganglia really is a complex network where parallel loops interact with re-entry loops, subcortical projections, changes internally in the excitability of the nucleus, so that the excitability of these cells actually is very variable and generally highly asynchronous population activity, where firing rate, firing pattern, coupling, uncoupling, oscillatory activity, all that comes together to produce a sort of code of activity. And I'm just showing this just to sort of make you think that we should not be so puzzled. At least I shouldn't be so puzzled after this lecture, because it is so complex. So why should we expect that we were going to understand tremor, rigidity, and akinesia just by one single mechanism? There must be many mechanisms. There must be many ways whereby these different pathways and different functions, different ways of functioning, produce symptoms. Anyway, the idea is that, and my prediction is that in the near future, we'll be able to pinpoint a specific circuit and a specific neuronal patterns of activity to understand these major manifestations. I can tell you that with David Marsden, years ago, we were well aware of the complexity of the basal ganglia, and we play through planes, hotels, with these drawings, exchanging these ideas here and there. Here I tell him, need some more inspiration to decide what the GPE does to come. Or here down we could say, Buenos Aires will give me passion to understand, things like that. And this was part of the game. And of course, one of the major interests for Marsden was the paradox, those things that we couldn't understand. The one regarding how pallidotomy eliminates dyskinesia is really being very discussed in many events, and I'm not going to deal with this because it's been well published in many occasions. So I'm going to concentrate on this apparent impunity of the basal ganglia to ablation, because after all, if you have any structure and you destroy it, one would expect that there would be some kind of consequence. It just doesn't make any logical sense that one destroyed a structure and all what you get is improvement. What was the function of that structure that has been eliminated? So this notion, we discuss it at length in this paper that continues to be a classic, and I have studied in detail in a number of patients between La Habana in Cuba and Pamplona, where patients who have extremely asymmetrical Parkinson's disease with dopamine depletion in the posterior putamen. We did a lesion in the subthalamic nucleus. The patients had hemichorea, hemiballism, that occurs in 8% exactly of all patients, but in those 8%, the dyskinesia does not go away, does not stop, so we needed to do a pallidotomy to eliminate the involuntary movement. So in effect, this patient was heavily dopamine depleted on that hemisphere in the striatum, has the STN block for the indirect circuit, had the direct circuit also blocked because of the pallidotomy, and one could say that the basal ganglia in that hemisphere was not working almost at all. Nevertheless, he actually did very well. Okay, I have here cut off part of the window, so I don't see, I have to look at here. So what happened is that he performed very well. The right upper limb mobility improved a lot. He didn't have any problem after we got rid of the dyskinesias with the pallidotomy, and careful analysis by Marjan Jashanhasi and John Rothwell in Queen Square, we saw by and large better results on the operated side, except that the patient could not learn, didn't perform well with new implicit learning. In other words, when he has to acquire new information implicitly that was not explicit to him with the affected side, that was wrong, was very, very wrong, was very affected. More recently, Thomas Bureau in Bordeaux has done an experiment in 2 monkeys. Monkeys learn a manual motor task. Then they did bilateral GPI blockade with muscimol in the GPI. And what happened is that the monkey, again, like in the patient, the motor learning that had been previously established, they performed well, but they couldn't learn new motor learning. In other words, the same like in the patient, what was already there could be performed, but what wasn't there couldn't be, was defectual. And a very old study from Peggy Anderson and Bob Turner is actually very interesting. They record neuronal activity in the GPI and in the thalamic receiving area, so that they were sure that they were recording in the projection. Then they trained the monkey to do a motor task, and once they has learned, they block the GPI and record and repeat the task with the GPI block. And see, what happened was that the task repeated after blocking, there was no major motor deficit. The monkey did well, performed well, despite having no GPI, and thalamic neuronal activity did not care that the afferent from the GPI was not there. They increased their baseline firing rate, but they continued to respond perfectly well in relation with the task. When the monkey started, stopped, did the sequence, etc. In other words, these thalamic neurons continue to work, to function, to fire exactly as normal, despite the fact that they didn't have the GPI influence coming from the basal ganglia. So altogether, one would say that this experience in patients and monkeys would suggest, just by pure logic, that the basal ganglia is not needed for performing habitual routine learned tasks. That obviously goes tremendously against what my maestro, Marsden, put forward in this magnificent and famous lecture. The basal ganglia deals with the automatic execution of learned motor plans. Now, Marsden was rarely wrong. In fact, he was right almost all the time, even for the wrong reasons. So to think that this is wrong makes me nervous. So I come around to see what is behind this. The essential element of this possible new paradox are performing routine, learned or automatic tasks, this is all the same, is mediated by the motor circuit, which is the region of the striatum that receives the nigrostriatal projection from the lateral nigra. This is here in the monkey from Hubbard, ventrolateral neurons projecting to the motor putamen in the most caudal part of the motor area of the frontal cortex. The more you go rostral, the more you go into medial substantia nigra and more anterior in the striatum for associative, cognitive, decision-making, and limb emotional components. Performing a habitual or automatic task activate the putamen, as predicted, just here, this part of the motor circuit, while when you are in goal-directed, which is when you are not working in automatic mode, when you have to think, when something is new, when you have to take decisions, when everything is not predictable, then the activity in the basal ganglia goes anterior. Therefore, this is the system that deals with automatic movements. Now, in Parkinson's disease, the region which is most vulnerable to and hit by degeneration is the ventrolateral tile of the nigra. This beautiful picture given to me by Professor Halliday shows this wonderful cell loss here compared with this normal brain of a patient, showing exactly where these cells are lost, giving rise to this dopamine loss in the posterior putamen and the reduce and the impairment of automatic movement. So all these fit. If you lose dopamine in the sensory motor region of the nigrostriatal system. Automatic, habitual tasks are first impaired because this is what is this is what they take care about. To lose neurons there has to be a number of vulnerability mechanisms. And for instance, it's been shown that one single axon in the rat arriving to the striatum arborizes and makes something like 200,000 synapses for the sensorimotor striatum, while in the limbic striatum it does an order of magnitude less, something like 20,000 synapses. So it's been hypothesized that all these non-stressors like mitochondrial, calcium influx, protein, oxidative stress, etc., may be acting through a system which is under stress because of high energetic consume, as put by Paul Bolam in this paper in movement disorder. If you are under such energy stress, many of these stressors will be harmful and will impair the physiology of the system and perhaps cause the degeneration. But the problem here is that if performance of an acquired action doesn't rely anymore on basal ganglia output, and therefore the motor basal ganglia circuit is not activated, why is then that these neurons are going to be overactivated if we don't need them? So I see here a major puzzle and paradox. So how am I going to be out of this? Classical studies by Wolfgang Schulz in the 90s have shown that dopamine cells in the nigra only fire, as shown here, in response to reward and not to movement. And that, of course, supported this idea. But in fact, this has changed radically recently. And here is a study in the monkey by Matsumoto indicating that VTA cells do respond to reward or don't respond, do the opposite, silence with a noxious stimulus like an air puff, while the lateral dopamine neurons in the nigra respond equally to either stimulus. They worked this out and they show that the lateral substantia nigra actually shows responses to a number of situations, relations with motivation, salience, orienting, cognitive processing, etc. And this could be a sort of the dorsolateral, not the ventrolateral, but the dorsolateral. But this, to begin with, modify the idea that substantia nigra cells don't fire in relation with activities in the external world. Therefore, one would have to conclude that they are more frequently activated by different stimulus and tasks. Here, now I have a problem here to find the, okay. This is a study, very important study from Rui Costa in Lisbon. The mice learns to do a sequence of activity going from one point to the other to find the reward. The reward is given randomly, so he cannot move according with the reward. He has to learn the sequences, and once he learns the sequences, he will continue to do that regardless when the reward will be given, which is the experimental definition of a habitual task. So those mice learned the task, and then it was shown that neuronals in the substantia nigra fire physically in relation with the beginning or the end of each of these sequences of movement, and they were here in the lateral substantia nigra, and also neurons in the striatum also fire at the beginning and at the end of the sequence, and these were neurons mainly in the sensory motor striatum. Therefore, self-initiated habits elicit phasic dopamine responses. More so, and recently, in this study, have shown that, in fact, the mice shows a kinematic direct relationship between acceleration of the head and spiking activity in the nigra. so that a very straight, highly significant line could be built between the movements, the acceleration, and the spiking, either for the cue to move or the actual reward. And the authors claim that they have therefore disengaged the reward part of the self-response with the actual kinematics of the response. So one could have here a situation whereby this new cortico-nigra projection can modulate neuronal activity of the nigra. We have our all well-known direct cortico-subthalamic nigra projection also, and the substantia nigra is no longer isolated. The sensory motor substantia nigra is no longer isolated from the basal ganglia and is actually overactivated probably most of the time. Therefore, and this is schematic, speculative, imaginative, when we do a routine motor task, we are stimulating our motor circuit and our lateral substantia nigra neurons. When we do something more cognitive, associative, this is the circuit, and here we stimulate both. We recruit the more medial neurons, but these also continue to fire when the situation is emotional, then the accumbens and the limbic, but also the motor part of the nigra. In other words, whichever we do, according with this proposal, these cells in the nigra ventrolateral, the ones who die in Parkinson's disease first, are activated and recruited. Even more, it is now known that we learn from goal-directed to habit formation, and that is this switching another important task of the basal ganglia and the nigrostriatal system. But it's also the case that we have a system for inhibition, and we have goal-directed inhibition and habitual inhibition, which probably activate the same corticobasal ganglia circuit as the active, the moving part of goal-directed and habitual activities. So this is just totally imaginative, but I would have thought that it's highly likely that dopamine cells are also engaged in the formation and in the monitoring of habit in terms of inhibition, not just habit in terms of action, giving another loading to the system. So a wide range of events activate sensory motor dopamine neurons. They correlate with movement kinetics. Automatic habitual tasks engage sensory motor dopamine response, and with Ledia Hernandez and Pete Redgrave, we're doing studies now in the rodents to show that indeed all these features are risk factor for cell vulnerability in Parkinson's disease. Indeed, I think these cells, the ventrolateral nigra cells projecting to the motor putamens, die because they are over-engaged. And my prediction is that the more things we do in our new world, the more they will engage and the more vulnerable they will be. So just to wrap up, there are several points which remain to be discussed. Why do patients tolerate lesion of the basal ganglia so well? I can say that they tolerate it, but not that well. If we look carefully, they do have errors, like inhibitory errors, defectual errors to do simultaneous activities, etc. So this is not completely crystal clear. Primates in those experiments have been overtrained to do one thing, and there is nothing else they have to do when doing the experiment. Therefore, the need to use the basal ganglia in those circumstances is low, and therefore this could explain why they perform much better. More worrying to me as a clinician is that while one could understand and concede that akinesia is the pathological counterpart of movement control, what is the physiological meaning, if you wish, of rigidity and tremor? What is it? What would it be? What is rigidity telling us in terms of motor control? What is tremor? Is tremor just a noise, or is revealing some kind of physiological mechanism? That has to be understood. And coming to my primary interest, if multitasking leads to functional overload of the nigrostriatal system, how can the focal asymmetrical motor features of PD at onset be explained? This leads to intriguing daily life features, such as the risk of Parkinson's disease will be influenced by features such as predominant body part use, for instance, hairdressers versus professional cyclists, or task versus multitask, dependent occupation, an assembly worker versus a designer, or differences between people who are focused and do one thing at a time versus those who are constantly engaged in several activities simultaneously. Those are questions that may be out there and that one needs to try to find out, and these are just ideas. So I finished my lecture with a proposal that we put forward that the nigrostriatal projection, and in particular the sensory motor region, is engaged in the control of learned habitual behavior, a demanding function during simultaneous and multi-domain action. This, in turn, is a major vulnerability factor for the ventrolateral neurons of the substantia nigra ventrolateral region in Parkinson's disease. This is my new center in Madrid. I'm part of the group, growing as it is. Ines and Raul have worked a lot with this PowerPoint, and Ines is making experiments in the monkey to prove some of these ideas. Pete Redgrave in Sheffield is like my guru, and Maria Rodriguez-Oroz, my former colleague, continues to help. And I can't think any better way to finish than by reiterating this writing that David Marsden wrote, because those papers that I wrote with him, of course, he was the one who knew English, as you can easily understand. So we can say that no doubt our difficulties will be explained by future knowledge and our speculations will be disproved. But we hope they will spur others to make the discoveries that will explain the paradoxes we find so difficult and that will disprove our theories put forward here to tell us really what the basal ganglia do, to which I like to add and why neurons die in Parkinson's disease. Thank you very much.